for inviting me to talk about this topic, which is leukodystrophies and autopsies, which is very close to my heart, because I realized during my almost 25 years of career how important it is to study the human diseases in the patients. So also in the patients, if they donate their body for research after that. I will share my screen. You should see it.
Yes.
Good. Okay, so we can start. This is me again. I have nothing to disclose. Okay, these are the learning objectives of this webinar. I hope that I will convince you of how necessary it was to articulate a new definition of leukodystrophies based on the new knowledge of these diseases in patients and the new achievements in basic research on the brain. I hope that I will let you see how it is possible to figure out in the brain of a child with a leukodystrophy which cell types are the one driving the disease. So helping address all the experiments that are needed to understand the disease mechanisms. And also, I hope that I will show you how neuropathology, so the study of the patient's brain, can help you understand how treatment works and how treatment may fail. This is the outline of the webinar. I will take you through the concept of leukodystrophy, how it developed in the last 50 years. I will show you how neuropathology can help evaluating the effects and the failure of treatment. I will show you in another example of a disease how neuropathology can help explain the disease mechanisms, and then I will conclude and thank everybody. Okay, this is a very simple cartoon depicting our brain. And you see here in the middle, there is a coronal slide of our brain, and you can see that there is some color stuff on the outside, that is the gray matter, and some white stuff on the inside, and that is the white matter. And the leukodystrophies are the genetic diseases of the white matter. If we look at the white matter under the microscope, we have a lot of different cells. We have the cell processes of the neurons that are allocated in the gray matter. We have the cells, the oligodendrocytes, that make an insulating substance around the neurons to improve the quality of signal transduction. But we also have a number of other cell types like astrocytes and microglia that help maintaining the homeostasis in the brain. Now, when I started in the 80s studying the leukodystrophies, the definition that was given was very strict, and it was given around 1985. And it said that leukodystrophies are genetic and always progressive disorders that affect the myelin, so the insulation of the axons, of the cell processes of the neurons, either because the myelin is not deposited properly during development or because the myelin is lost later. So it was a concept that really was focused only on myelin. But you have to realize that at that time, almost no gene defects were known about the leukodystrophies. MRI had not entered the clinical practice, and the few data available were available for the 150 years before, in which neuropathologists looked at some brains of patients and could recognize some disease, especially from the biochemical standpoint. This had a very important, intellectual consequence that at that time, and I have to say up for the next 25 years, and there is still people thinking it at this time, people thought that the only curative treatment for leukodystrophies was to stop the loss of myelin and to restore the myelination in the brain. Now then in the 80s, MRI was introduced in the clinical practice, and it became very clear that the MRI tool, the imaging, is extremely sensitive for white matter abnormalities. And you can see here a normal brain of a child, and you see that the white matter in this sequence is very dark, while in these examples on the right side, the white matter of the brain becomes very white. So it's completely affected. And it was so easy to make a diagnosis, a generic diagnosis of leukodystrophy using MRI that imaging completely replaced the neuropathology. In the 80s, end of the 80s and the 90s, the Amsterdam group, led by Professor Jaap Valk, who was a radiologist, and Professor van der Knaap, who is a child neurologist, expert also in radiology. She's going to give us a webinar on leukodystrophies again in September, developed what is called the MRI pattern recognition. That means that they used MRI to make the diagnosis of leukodystrophies that could be confirmed mainly on biochemical basis. But they thought if we can do this with leukodystrophies of which we can identify the defect underneath, maybe we can also group undefined leukodystrophies and then study them to reveal what the defect, the causing defect is. At that time, genetics was taking in. There was very complicated way to find out in very large groups of patients what the gene involved in a disease was. But in the last, I would say 10 years, more or less, a new technique was introduced now in diagnostics too, since 5 years, about more or less, which is called the next generation sequencing. And this way of looking for genetic defects allows to have very small groups, even single families in which one sick, one healthy child and two parents are necessary, are enough to make a genetic diagnosis. So by using this tool, it became soon very clear that most leukodystrophies are actually not due to diseases of the myelin or the myelin-forming cells, but are due to defects in genes that are specifically expressed in other cell types than the oligodendrocytes. In the meantime, the basic research was going on. People were asking themselves, how is myelin formed in the brain? How is it maintained during adult life? And is there a possibility to repair a myelin damage? This is again a cartoon of the cellular components of the white matter. You have again the axons enwrapped by myelin, and you have all the other cell types. Now, if we look just at the myelin-forming cells, the oligodendrocytes, researchers found out that our brain, even the brain of the older people, still contains a pool of stem cells of oligodendrocytes that continuously reproduce themselves, divide, to give rise to new myelin-forming cells. And this occurs through a process of maturation and differentiation of these cells until they become fully mature and able to enwrap an axon with the myelin. But what is more interesting that came out in the last 15 years is that the molecular mechanisms that underpin and regulate this process of differentiation and maturation of the oligodendrocytes are not that much intrinsic to this cell type, but are rather due to signals that come from other cell types. For example, in the newborn brain, if an axon starts conducting a brain electric impulse, at the same time, it instructs the oligodendrocytes to become mature and myelinated in order to have an optimal way to conduct that nerve impulse. And this holds true for all cell types in the white matter, including the astrocytes, the microglia, other kind of glia, and even the blood vessels. And this is a very complicated how can I say, molecular regulation of myelin development and maintenance that also regulate myelin repair after injury. But it has an obvious consequence. If the myelin itself, the oligodendrocytes making the myelin, are not the only one who regulate the making and the maintenance of the myelins, and if it's true that other cell types are necessary to optimally maintain this process, then it means that leukodystrophy cannot be only diseases of the myelin. So some years ago, we introduced a new definition of leukodystrophies, by which they are genetic disorders, not necessarily progressive, because some of them resolve spontaneously over time, that are due to a defect in any of the white matter structural components. And as always happens after a new definition, we also had to put out a new classification that recognizes, of course, the diseases of the myelin and the oligodendrocytes, but also the leukodystrophies that are due to the other cell types, the astrocytes, the interaction between the myelin and the axons, the interaction between the myelin and the microglia, the immune cells in the brain, and also the vascular system. Now, I will show you now a classic leukodystrophy. Oh no, we have a question. So Sanya has to kick in.
Exactly, here it is. You can vote and then...
Yes, what is a leukodystrophy? Possibility number one, a genetic disease of the brain white matter due to defects in any of its structural components. Or a disease only of the myelin. Or a disease only of the oligodendrocytes. Or a disease only of the neurons.
Yeah, so 60% have already voted, and I will now show the results.
Okay, well, I convinced you. Thank you, guys. Okay, then we go on. I will show you now. Yes, this disease. This is one of the diseases that was diagnosed biochemically already 50, 60 years ago. It's called a metachromatic leukodystrophy, and it's one of the most prevalent diseases in Europe. It is a genetically determined disease due to what we call lysosomal storage. Okay, lysosomes in our cells are the waste bins. So what they do is that they contain a number of substances, a number of enzymes that degrade waste substances, waste debris. And if this does not occur because the genes encoding for these enzymes have a mutation, then the debris, the waste, accumulates, becomes toxic, and in the end it kills the cells. There are different types of metachromatic leukodystrophy depending on the age at onset, and this is their typical MRI pattern. So you see that the white matter is diffusely white, so it is diffusely and symmetrically abnormal. So it's a leukodystrophy, and you see in this coronal image that also the white matter of the cerebellum is affected. Now, this is the metabolic pathway leading to cell death. This is the enzyme that should work, where the red arrow is placed, encoded by the gene that is mutant. Sulfatides cannot be degraded by this enzyme that doesn't work. So accumulate in the cells, becomes toxic, and kill the cells, especially the oligodendrocytes. Here you have on the left a normal brain, how it looks when you take it out of the skull, you fix it, and then you cut it again in a coronal way. And you see that you have again the gray matter on the outside. The white matter is very white. On the right, we have the brain of a child who died at 7 years of age of a metachromatic leukodystrophy. You see that the white matter has another color, is sort of yellowish and sunken. And you also see that the lateral ventricles in the brain are enlarged, meaning that the white matter is atrophic. If you look at this disease under the microscope, you should see a lot of myelin, which in this staining should be colored in blue. And you can see immediately that there are only few blue strands in this image. What is overwhelming is the appearance, the accumulation of cells that are pink. These are periodic acid Schiff colored cells, and these are the macrophages that enter into the brain to clear up the storage disorder. However, this clear up is not enough, so they accumulated the storage in the lysosome that here appear in pink, and they, in the end, they die altogether with the oligodendrocytes. Now, in the Netherlands, the current treatment for metachromatic leukodystrophy is hematopoietic stem cell transplantation. This means that a donor is chosen based on suitability of an affinity with the patient. Stem cells from the bone marrow or from the blood are isolated, expanded into a flask, and re-injected into the patient. Now, when we started looking at the effects of treatment in this disease, it was not really clear why this treatment could or could not work. The idea was that the cells that were transplanted in the patient were going around in the bloodstream, arrived in the brain. They were able to cross the brain walls, the blood vessel walls, enter into the brain parenchyma, and then release the enzyme that the brain of the patient was not able to make. And this enzyme would be picked up by other cell types and allow them to dispose of the storage waste. What was observed at that time is that actually there is a number of patients, a proportion of patients. These are mostly patients that are pre-symptomatic or pauci-symptomatic when they are treated, in which the therapy seems to work. Because you see here on the left, a patient before transplantation, you see the white signal in the white matter demonstrating the leukodystrophy. And after almost 5 years, the white signal change has almost gone. But there are also patients in which the disease progresses. And in these patients, what is remarkable is that over time, also the gray matter becomes affected. The cortex becomes very thin and the basal nuclei become smaller. So we started collecting brains of patients who died after treatment to try to figure out how the treatment worked. And this is again on the left, a patient of the brain of an untreated patient. You see the white matter that is all yellowish and sunken and affected. And here on the right, you have the brain of a treated patient with transplantation. And here you see that the white matter is nice and white with a nice consistency, and the lateral ventricles are not enlarged. So we could confirm macroscopically what we had already seen on the MRI, that there is a subset of patients in which transplantation actually slows down or even halts the leukodystrophy. But then what happens? How does this happen? The first thing that we asked ourselves were if the donor cells, so the transplanted cells, are really able to enter the brain. And we made a study using a very easy histological staining that stains for the sulfatide. And it stains for the sulfatides in red, and it stains for the normal macrophages in brown. So the macrophages, the cells that are full of storage of waste, toxic waste, remain red, while supposedly the cells that come from the donor are beige, yellowish. And you can see that in this slide, you have both kinds of cells, even the yellow one. So it means that the donor yellow cells are able to go around the bloodstream, past the blood vessels in the brain, and engraft the brain parenchyma. Then we looked if these cells actually of the donors actually contained the enzymes that is lacking in the patient. So here you have a low at the upper and a higher magnification image of the brain white matter of a donor of a normal control stained for the enzymes that is lacking in metachromatic leukodystrophy. And you see that all the cells contain these very little brown dots that are the lysosomes full of enzymes. If we look at untreated patients, the enzyme is completely lacking. But if you look after transplantation, the enzyme is back there. So the donor cells are able to reach the brain of the patients, but are also able to maintain a high amount of enzyme to help the brain survive. But now, how does that occur? We looked, the idea when we started was that the enzyme would be released by the donor cells, picked up by the patient cells, so it would have to be found back into the patient cells. So we did double stainings for with antibodies against the enzyme and against antibodies against proteins that are typically for the different kind of cell types in the brain. We did it for microglia up, for astrocytes here on the left down, and for oligodendrocytes, so the myelin forming cells, down on the right. And here the enzyme is depicted in green, and you see that actually the enzyme remains into the cells of the donor, is not picked up by the patient cells. So how is it possible that the white matter remains relatively healthy if these cells do not pick up the missing enzyme? There must be something going on. It's not an enzymatic correction, but there must be something else going on. If you look again under the microscope, what you see on a non-transplanted on the left and after transplantation patient on the right, what you see is that on the right image, there are a lot of cells, a lot of blue dots. These blue dots are the nuclei of the oligodendrocytes, the myelin forming cells. So this means that whatever the transplantation does, it does allow these cells, these oligodendrocytes, to survive. While you see that if the patients are not transplanted, these cells are virtually absent. Then we ask ourselves, okay, are these cells surviving, but are they also able to make myelin? And we used electron microscopy to find that out. And we looked for signs of myelin repair. Now, when a myelin sheet is made during development, it's quite thick. When it's made afterwards in life, in the context of a repair mechanism, is much thinner. And you can measure the ratio in between the axon diameters and the thickness of the myelin, and then plot them on a graph. And you actually see that in the MLD patients, the pink line is parallel but completely separated from the blue line. The blue line are the normal controls. The pink lines are the transplanted patients with metachromatic leukodystrophy. This proves that the transplantation corresponds histologically with repair of myelin and remyelination. But again, this cannot be explained by the fact that the myelin forming cells have the enzyme, because I showed you before that they don't have it. They don't pick it up. So we went looking for something else. Especially, we went looking for the inflammatory environment in the brain. Metachromatic leukodystrophy is a disease in which there is a lot of inflammation because there is a lot of waste accumulating and the myelin is breaking up, and that creates a pro-inflammatory environment in the brain. So we asked ourselves, is it possible that transplantation changes the environment into an anti-inflammatory environment? And this is exactly what we found. Using different markers for pro-inflammatory or anti-inflammatory cells, we found that after transplantation, the density of pro-inflammatory cells goes down, while the density of anti-inflammatory cells goes up. And why is this important? Because the anti-inflammatory cells, the anti-inflammatory macrophages, are able to secrete growth factors and substances that maintain the homeostasis of the brain. They are able to eat up all the myelin debris that is made, and that is prerequisite for repair. As long as the waste material is around, it cannot be any repair. And they also support the oligodendrocyte differentiation and maturation, helping them becoming a myelin-forming cell. But so this treatment, hematopoietic stem cell transplantation, may repay the leukodystrophies. But then how come that patients, their cortex become thin and patients become demented after a number of years that they have been transplanted? To figure out this, we looked in the cortex. And this is again the staining for the enzyme. You have in the upper panel the cortex, in the lower panel the white matter, and you see a control on the left side, and you see that all the cells contain the brown dots of the lysosomes containing the enzymes. If the patient is not treated, no cells contain the enzyme at all. But if the patient is treated, you see the brown in the white matter, but you don't see it in the gray matter. And we confirm this also doing fluorescent studies. Here the neurons are depicted in red, and the enzyme is depicted in green. And you see on the right panel that even if the patients are transplanted, the enzyme cannot reach the cortex. So the neuronal disorder goes on, is lower than the white matter disorder, but it goes on, and over time the cortex degenerates and the patients become demented and die. So we had to find another way of treating this patient to ensure that the donor cells also reach the gray matter in order for this patient to be globally treated and not treated as far as only the leukodystrophy is concerned. A question: does hematopoietic stem cell transplantation always work in metachromatic leukodystrophy? Never. It has no effects. Seldom. Only in the cases that are more advanced, meaning more severe. Sometimes, and often only on the white matter pathology. It always helps for everything.
Yeah, half of the audience has already voted. A couple of seconds more, and then I will show the results. Okay, and here are the results.
Yes, exactly. It sometimes it helps. We still have to figure out why, but only on the white matter pathology. So we probably have to change it. We have to figure out why these macrophages from the donor don't enter the gray matter. Is it a problem of the donor cells? Is it a problem of the gray matter that doesn't call them in a way? Is it a problem that we must give more enzyme? So combining transplantation with gene therapy, for example, so make donor cells in which the amount of enzyme is 2, 3, 4, 5 times in excess. We don't know that yet. But it helps understand what the treatment, the effects of the current treatment are. I have another example. Let's give it a couple of seconds because of the connection. Yes. My next example is an example of a leukodystrophy which is due to a defect in astrocytes. Astrocytes are the cells in our brain that maintain the homeostasis and orchestrate the repair after injury. And they do this by orchestrating, controlling a number of functions that are depicted in this cartoon. They control myelin, they control the plasticity of the synapses, they control the homeostasis of waters and iron, they respond to injury, and they control how substances go inside, from inside the blood vessels into the brain and get back in the same way. The idea behind these kinds of diseases, this kind of leukodystrophy, is that the normal astrocytes that we have in a normal brain, in a leukodystrophy is pathologic. And this corresponds to the increased expression of some proteins and decreased expression of other proteins. But changing the proteins that are expressed or not expressed means also changing the functions of the astrocytes. So the physiologic good functions are lost in these cells, and the pathologic bad functions are gained. And then by a mechanism of communication in between astrocytes, but also communication with all the other cell types in the brain white matter, this ends up resulting into a leukodystrophy. The example I want to bring to you is vanishing white matter. Vanishing white matter is probably the most prevalent leukodystrophy, especially amongst the children. And it is a disease characterized by mutation in 5 different genes that code for the subunits of a big multimer, a big factor that regulates the translation of RNA into proteins. Also, vanishing white matter has a very typical MRI pattern. Here you have a sequence in which the white matter is completely white, so it's completely affected. But if you use another sequence, which is depicted down, you see that the white matter becomes progressively more and more gray. This means that the white matter disappears over time, and it's substituted by a sort of fluid that has the same signal features of the cerebrospinal fluid. You have in these pictures the lateral ventricles, and you see that they are full of CSF that is grayish, but if you look on the white matter on the side, the color is exactly the same. If you look at the brain macroscopically, you see that the brain the white matter has completely disappeared, and there are big holes on here, for example. The white matter is replaced by big holes. And if you stain again for a staining that should stain the myelin in blue, you see that all the myelin is lost, and the white matter is replaced by holes. Hence the name vanishing white matter. If you look under the microscope, on the left is a regular staining. You see that what is typical of this disease is that the white matter falls apart, and only little islands of tissue are left, in which the cell density is very high. This is a very damaged brain, and in a very damaged brain, you would expect a very florid reaction of repair from the astrocytes. And if you look on the right, the brown cells are the astrocytes, and these astrocytes are very few, very scarce in the brains of these patients. So it's a disease in which you have loss of all white matter structures. The cells that are maintained are the oligodendrocytes. The astrocytes do not repair, but there is no selective disease of myelin. So this disease would not have been called a leukodystrophy in the 80s. Now, the process of reactive gliosis is fundamental for our brains. These astrocytes sense really the kind of injury and the entity of the injury. They change their transcriptional profile, they become bigger, intermingle their cell processes, and if there is a very severe injury, they build a scar around it. So this is the process of reactive gliosis and glial scarring. And they do it participating, communicating with all the other cell types in the brain. But the astrocytes are the one more or less orchestrating the repair process. If you look on the left, you have vanishing white matter. If you look on the right, you have the astrocytes, again in metachromatic leukodystrophy, a disease in which reactive gliosis occurs normally. And you see how the density of the cells is different. We would expect a similar number of cells, also in vanishing white matter, but this does not absolutely happen. Now, reactive gliosis, I told you before with the myelin that oligodendrocytes make the myelin only when they are mature cells. Reactive gliosis is the same for astrocytes. Only mature astrocytes can make a process, orchestrate the multicellular response of reactive gliosis to injury. So we asked ourselves, is it possible that in this disease, the astrocytes are there, but they are too few, and they're maybe not mature enough to work properly? So we went looking for it with immunohistochemistry, and we used many proteins, many antibodies against markers that are typical of immature astrocytes. And we found that in this disease, the astrocytes had the same immunohistochemical profile of the astrocytes in the brain of fetuses. Now, typical of the brain of fetuses is that the astrocytes are immature, and if you have a damage like a stroke or a trauma, you will get a hole, you will not get a scarring. And this is exactly what happens also in vanishing white matter. But again, so we can say the astrocytes don't work because they're immature, but what happens to the oligodendrocytes? There is no myelin again, but the oligodendrocytes can proliferate like crazy. Look at this image on the left compared to a control on the right. The number of blue dots that are the nuclei of the oligodendrocytes is like 4 or 5 times too many. Nonetheless, they cannot make myelin. So we argue again, is it possible that they don't make myelin because they are too immature to make myelin? And we stained them and found them that they express the same markers of oligodendrocyte progenitors that again they do in the fetal brain. So astrocytes and oligodendrocytes in the brain of vanishing white matter patients are like the ones that we see in the brains of fetuses. But then, okay, 2 cell types are heavily affected. They can explain the MRI. They can explain the kind of degeneration that we see under the microscope, but which is the chicken and which is the egg? Now, at end-stage disease on the brains of the patients, you cannot see that. You have to play around a little bit. The only thing that we could see in the brains at end stage of the patient is the amount of substances made in the fetal brain from astrocytes that maintain the brain liquid for the other cells to migrate properly. And we found that these substances, one of which is hyaluronic acid, all the ladies know hyaluronic acid because it's in creams, and it helps to maintain the skin very hydrated because it collects and retains water. Well, this hyaluronic acid in the brain of these patients is 800 times more abundant than in the brain of the normal people. So the white matter is really prone of becoming full of water and very fluid. But this tells us again not who has the culprit, the astrocytes or the oligodendrocytes. To do this, we used mouse models. So we took astrocytes and oligodendrocytes from the brains of wild type and vanishing white matter mice, and we plated together them in a culture dish. And then we found in different combinations, and then we looked if the oligodendrocytes were able to make myelin. And we found that if the sick oligodendrocytes are cultured with the wild type astrocytes, they are able to mature and make myelin. But if the wild type normal oligodendrocytes are cultured with the vanishing white matter astrocytes, they completely stop growing and are unable to make myelin. So at least as far as the myelin production is concerned, this is not an intrinsic problem of the oligodendrocytes, but is secondary to the astrocytic pathology. And the astrocytes in the white matter are not the only ones that are affected. We have found affected astrocytes in the cortex of the cerebellum, the Bergman glia, but we also found that the astrocytes are affected in the retina of these patients. So in the top, these are white mouse retinas, but you see the same on patients. In the top figure, you have a normal retina with a nice lamination of the neurons. And if you look in the lower panels, in different kind of vanishing white matter mice, you see that the lamination is completely lost. This is a form of retinal dysplasia that accounts for part of the visual loss that patients with vanishing white matter have. And you can figure it out also in vivo by you doing a sort of electroencephalogram of the retina, in which a peculiar part of the trace, which we call the B wave, is affected because of the affected astrocytes in the retina. And besides this, in very young children, also glia-like cells that are spread out of the body, in the lungs, in the liver, in the pancreas, in the guts, are affected. And these children also have involvement of other organs besides the brain. So vanishing white matter is a disease of the astrocyte, of the oligodendrocytes, so the myelin forming cells, so a classic leukodystrophy. A disease of the astrocytes, a disease of the microglia, the brain immune cells, or is it not a brain disease?
Okay, 10 seconds to go, and I will show the results. Okay, so here are the results.
Yeah, well, it's a disease of the oligodendrocytes and the astrocytes, but it appears that the astrocytes are driving the disease of the oligodendrocytes. So the first two questions, two answers are equally correct. Thank you very much. We can proceed. So the key conclusions. I hope that I convinced you that the definition of leukodystrophy had to be revised, because the knowledge on the genetic defects of leukodystrophies and the knowledge of the biology of the white matter increased so much that they demonstrated that the myelin and the oligodendrocytes are not the only cell types that are affected in this disease. I hope that you agree with me that we can now, with what we know at this time, we can call leukodystrophies all the genetic disorders in which any white matter structural component is primarily affected. And I hope that I have convinced you that a new definition is necessary to understand, to better interpret, but also to better understand the complexity of the white matter. And I hope that you agree with me that nowadays, with what we know about leukodystrophies and white matter biology, when we think about treating patients with leukodystrophies, we need to repair more than myelin alone. Because, for example, if we look at a leukodystrophy with astrocytic influence, in which there are many progenitors that are unable to repair, to form the myelin, if we repair the myelin, the oligodendrocytes are not able to survive anymore, and the leukodystrophy is not going to be repaired, or even is going to come back. Now, last question. Does neuropathology help unraveling the leukodystrophy's disease mechanism and the effects of treatment? Absolutely not. autopsies are useless. Often, autopsies, however, are too much of a burden for families and doctors. Always, autopsies should always be performed. I don't care.
Okay, half of the attendees have already voted and 10 seconds to go. Okay, here are the results.
Oh, okay. It is true that autopsies are often a lot of burden for the patient is dead, poor guy, but for the families and for the doctors. It is not easy for a doctor to ask for an autopsy, and it's not easy for a family to accept an autopsy. However, the most important thing when you propose an autopsy is to talk about the kind of information that it can reveal. It's to say that there is no in vitro model, there is no animal model that can reproduce the disease like it is in the brain of the human patients. That every autopsy is a sort of organ donation that helps unraveling and understanding more and more the diseases. So it helps treating and better diagnosis and better taking care of the patients that are still alive. Because unless we really understand what goes on in the brain of a patient with the leukodystrophies, with a rare disease, in the body of a patient with a rare disease, we will not be able to think of an effective treatment. I have one last slide. I want to thank the group of the Amsterdam Leukodystrophy Center, which is headed by Professor van der Knap. And we work on leukodystrophies from all standpoints. And I have on the right an old picture of my group. It was about 1/3 that it is now. And all these girls are really working a lot to unravel the disease mechanisms of leukodystrophy, to understand repair, and to figure out how to promote the brain possibility, intrinsic repair in the brain, to achieve a better care for the patients with these terrific, tremendous kinds of diseases. Thank you very much.
Thank you, Marianna, for this very instructive webinar. Thank you for telling us the whole story from the past until now and the state of the art of research in this field. This was very interesting, and we have some comments. However, it is from family member from Bulgaria. I would say, Susan, that it should be treated in another frame. Your question, maybe you can contact Marianna personally and explain your case.
Your case, please do that.
Yes, I have also shown the email address of Marianna at the beginning.
And Susan, I can also provide you with my telephone number and you can call me and we can have a chat about it.
Thank you. Yeah, and in the meantime, you can write other questions you have with connection to this, to the content of the webinar. This was also the last webinar before our summer break. In September, we are restarting again with our webinar series, and we have a related subject, vanishing white matter, and it will be given by Professor Mario van der Kaap from the Amsterdam Leukodystrophy Center and Medical Center. Okay, so
we have another question in the chat. If you download the webinar from the slides, you find my email, so you have my contacts.
Exactly.
There is another question: is contrast enhancement possible in leukodystrophies? It is, only in a few of them in which the blood-brain barrier is heavily damaged. But we just did a histopathology study on the brains of the patients with different kinds of leukodystrophies, and we found out that this blood-brain barrier, more or less, because one cell type or the other, is always affected in the leukodystrophies. The only thing is that the contrast enhancement is a big, the molecules, the gadolinium used for contrast enhancement is so big that it only passes the barrier if the barrier is completely damaged. So in leukodystrophies, there is also an involvement of the blood-brain barrier, but is often very subtle. What we gonna do if we haven't any research and genetic testing? Okay, what you can do is to get in contact with us, collect blood, send the blood for us for genetic testing. We have a sort of panels of different genes that can be looked at. The genetic testing is often very much addressed by the MRI picture, because many leukodystrophies have a prototypical constellations of signal changes on the leukodystrophies. And if you have the possibility to do an autopsy after a patient passes away, we are always available to receive the brain and study it together with you and see what, and of course, in case we find something new about that disease or about the leukodystrophies in general, publish it together to make it available to the whole scientific community.
Great, thank you. Are there any more questions? You can ask them by posting them in the Q&A box. It doesn't seem so.
No.
Okay, so thank you again, Marianna. Thank you,
you,
everybody. And hope that everybody is fine and have a nice summer break, and see you soon in September 13th for our next webinar. Bye-bye.
Have a good day. Bye-bye.